Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the molecular basis of inheritance. We're going part 3, packing of DNA helix. Taken the distance between as in a two constitutive base pairs around as in a 0.34 nanometers. So before only we discuss when you take as in it the two base pairs, when you see as in two base pairs, in this distance how much? In this distance around as in 0.34 nanometers. Yes, here the length of the DNA molecule, the helix, the typical mammalian cells get calculated simply the multiplying to the total the number of the base pairs with as in a distance between as in a the two constitutive base pairs. That is what they given as in a 6.6 into 10 to the power of 9 base pairs into how much? That is in a 0.34 into 10 to the power of 9 base pair. The around as how much? 2.2 meters. Around as an 2.2 meters. It is in a their human diploid DNA length. Next, it comes out to be as in a approximately 2.2 meters. The length of that for the greater than the dimension of in the typical nucleus, approximately they show as an 10 to the power of 6 meters. How such as a long polymer they packed in the total cell? Let us see here. Here, the length of the Escherichia coli DNA 1.36 nanometers can be they calculate the number of the base pairs of the Escherichia coli. So how to get calculate here? 1.36 into 10 to the power of 6 base pairs here into totally 3.6 into the 10 to the power of 9 base pairs. Am I right? Around as how much distance? 0.34 nanometers. Around as in a here 1.36 into 10 to the power of in a there are 6 base pairs we are gets. Next, in prokaryotic such as in Escherichia coli through they do not have a differentiated nucleus and the DNA is scattered through as in a cell. The DNA, the negatively charged, the held with as in some proteins that have a positive charge. The region that termed as a nucleotide, the DNA nucleotide is the organism is in a large loop held by as in a protein. See here, it is a complete loop-like structure. It is in a completely the loop-like structure is noticed here. The number of proteins they get arranged. So mostly saying about as what the DNA. So the DNA has in that negative charge and the proteins has in a positive charge. They have in a positive charge. And let us see this how do they packing when you observe it as in a prokaryotes as well as eukaryotes. Let us see in eukaryotes first we see. The eukaryotes, in that eukaryotes, this organism, the organization of in a the more complex. So when you observe as in a that entire the eukaryotic the nucleosomes they get arranged as in that complex behavior. It is completely they get arranged as in a their complex behavior. See, they are in a set of the positive charges as in the histones as present. So here I took as in a 4 histone proteins, that is in a H1A, H2B, H2 and H4. These are histone proteins as present. Am I right? The proteins are acquires the large depends upon the abundant of amino acids residues with as in a charge as in a particular side chain. So these are completely they having as what amino acid residuals as present here. Amino acid residuals also they are present. The histones as what the mostly the rich of the basic amino residuals present, that is in a lysine as well as arginine. That is what lysine as well as arginine. The both of the amino acid residuals they carry the positive charge in their side chain. So when this completely when you take here it is they are completely the amino acid residuals to get packed like only. Am I right? It is in a complete the packed rapidly they are packed to as in the histone proteins. Here the histones are organized to form as a unit of the 8 molecules is called as a histone atomers. See, when you take here in that front of, I draw only for 4. So front side they have in 4 and back side of the complete they have in a 4. There are 4 plus 4, how many total? 8. So that is called as what? Histone octamer. Octo means how many? 8. Here the negative charge of DNA wrapped around as a positive charge of the histone atomers to get structure is called as nucleosome. So it is that this total structure is called as what? It is called as a nucleosome. It is called as what? Nucleosome. So the typical nucleosome contain how many base pairs? 200 base pairs. It is contain how many? It contain as in the 200 the base pairs as present. Next, the nucleosome constitute the repeating units of the structures. The nucleus is called as a chromatin. See here, the repeating units are there coming here. The repeating units are called as what? That is called as a chromatin. Chromatin. Okay, and the thread-like structure scholars bodies is noticed that in the nucleus, and the nucleosomes, the chromatin, they are seen on the beds on the string structure when we go under the electron microscope. So when see under electron microscope, it is appearing their bed string on the structure. Means how do they get arranged at the points, and then one by one that structure you noticed under electron microscope. Okay, let us. The theoretically how many such as in a bed on string structure do you have image the present on the mammalian cells? When you observe as in how many, they are completely, see here the total, it is in a bed on string structure, it is in a bed on string structure, like that completely the chain like structure will be to get form in that in a mammalian cells. Next, the based on the bed on string structure, the chromatin the packed to form as in a chromatin fibers, they are in a further coiled, they are condensed the metaphase stage, the cell division to form as what? Chromosome. So after the cell division, what happened? This enter the chromatin to get forms as in a this kind of the chromosomes are forms. This kind of the chromosomes as form. Next, the packing of the chromatin, the higher level of require the additional set of the proteins. It is collectively called as what? Non-histone chromosomes. Then NH6 proteins. So, for example, you take as in one particular the DNA strand. In this DNA strand, here the information is not passed here to here. So, these parts are called as what? That is completely called as a non-histone complex. That is called as what? Non-histone complex. Next, after in this non-histone proteins when the present, what happened? The typical nucleus, some the regions of chromatin is loosely packed, the staining light that it refers to as an euchromatin. Loosely packed genetic material is called as an the euchromatin, and the chromatin is mostly the densely packed, that is stained dark is called as what? That is in a heterochromatin. Loosely packed is completely euchromatin, densely packed is called as what? Heterochromatin. The chromatin is said to be that is in a transcriptionally active chromatin, whereas heterochromatin is what? Inactive. That is what? Inactive. Okay, this is the in a packing of genetic material that is in a DNA. Remaining to discuss in next class. Thank you for watching. Like, share and subscribe.